All right, welcome to another AP Biology review video. As you can see, this one's going to be about cell communication. Now, the first 3 units were mostly setting the foundation of biology. We need molecules that will then form a cell membrane and cells to give us a body. And then you need energy in order to keep it all running. Now, this unit and the next one are kind of about how cells actually work, how they actually get things done. And especially in a multicellular organism, a lot of that comes down to cooperating and coordinating your actions with the other cells so that you can all know what you're supposed to do. And we went over quite a few examples of this. One thing that we talked about was, in this case, how a developing organism, this is from a worm, a tiny little microscopic worm called C. elegans, where this cell is telling this cell to become the intestines of the worm that is developing, right? And so, and same thing happens in any multicellular organism where the tissues have to form and grow in the right place for the right size. All these things need to happen. And so we need to talk to each other to do those things. In this case, this one is from the nervous system where one neuron is trying to tell another neuron what to do. Or in the largest scale version of communication in the endocrine system, hormones will be released by a cell that's, say, down here in your adrenal gland, which is on your kidney, and that needs to travel to your lungs and your heart and your leg muscles and your calf muscles and travel all through the body, right? These are different levels of communication. The first two would be called local signaling. And then the middle one, the hormone one, would be called long distance signaling, just because of the geographic location of where the signal originates and where it finally arrives at. Now, one of the things about science, one of the basic ideas of science is that there must always be a physical connection between phenomena. So we need a physical way for the cells to talk to each other, and it's all going to start with something called a signal molecule. Sometimes that's stuck on the outside of the cell membrane. Sometimes it gets diffused a short distance from one to the other, or like again, sometimes it goes through the blood. But there's always some sort of signal molecule, a physical object that's going to then interact with other physical objects. And this leads to the cell communication pathway, where we have to receive the signal. Then that is going to trigger a chain reaction of molecules to transduce or carry the signal further into the cell where something's going to happen, right? Doesn't matter what we're talking about. Other examples that we talked about in class were things like how the immune system recognizes pathogens, or how even single cell organisms need to communicate through something called quorum sensing, where individual bacteria will send out little molecules to let the other cells know how many cells are around, so they can launch a coordinated attack on whatever they're trying to do, right? Everything that's going to happen involves these 3 steps. So to get further into it, the signal is often called a ligand. A ligand is just a kind of generic term for some molecule that fits inside of another molecule based on shape. Each signal or each message has its own signal molecule with its own particular shape. This way you can keep your signals straight so that you can have different signals mean different things and not get them confused with each other. These cells have different protein receptors on them. So if you send a signal out like this one, that is only supposed to go to, say, your intestines will have the correct shape receptor, and this will make something happen. But you don't want your brain to freak out when your intestines are trying to freak out. So this cell and this cell, say maybe that one's in your liver, you don't want them to do anything. So they have the wrong shape receptor, and therefore they don't react when the signal gets sent out, right? And so through a combination of shapes, different shaped signals, and different receptor proteins on the outside of the cell, you can get a lot of different communication without getting confused. The The signal molecules kind of come in two different categories. And so here are two pictures of some molecules, which will bring us back to the first unit. The important thing that we care about mostly, as you might remember, are these polar or non-polar molecules, right? And so by looking at these, we hopefully can recognize that this part right here, including that little bit, is all H's and C's, hydrocarbons, which makes a very big non-polar section. I know it does have the OH's and the O, but this one has a lot of OH's and an N and two. There's more OH's, and so this one is polar and this one's non-polar, and that's important because the location of our receptor protein will change. Polar molecules can't get through the membrane, so they're going to have a surface-bound receptor, whereas the nonpolar ones can go through the membrane that's being shown over in this picture. The nonpolar one can just go straight through the cell membrane and get into a receptor that's found on the inside of the cell in the cytoplasm, whereas the nonpolar ones, which are the more common ones, they have the receptors on the outside of the cell, and that's something that comes up relatively often, so I want to make sure that that gets thrown in there. Right? So that's for reception. We have to get a molecule to connect to the receptor protein. There's lots of different receptors. The important part is the conformational change, that when the signal molecule binds to one of these different receptors, it's going to make them change shape somehow. This one is going to make it kind of change shape from that into that, which makes this little deal fly off. Down here, we have two pieces that are kind of separate, but when the signal molecules bind, they make the piece come together, and that makes the shape change. Or in one of the most like literal interpretations that I don't even know, it's not a metaphor or something, but this is like a door, right? We want a door to open. That's a change of the shape. And so when the signal molecule hits there, it makes the door open, and then stuff can happen. And then when the molecule falls off, the gate closes, the stuff's all reversible, right? You don't have to remember anything specific. There should always be a picture, or at least a detailed description of what's happening. But the idea is that the signal has to hit the protein, which makes the receptor change shape. Once that changes shape, we can now send the signal further into the cell towards where it needs to go, towards the response. This is going to happen with two basic steps. First one's called a phosphorylation cascade. Hopefully this kind of vocabulary isn't going to come up, but phosphorylation is something that happens a lot. It means that a molecule, a protein, is going to give a phosphate group to another molecule, usually another protein. We can also dephosphorylate things. Sometimes we can take that off. Again, you shouldn't have to memorize that, but you might see those words show up in questions. And so it's helpful to know what they mean. This is the phosphorylation cascade diagram, right? We've already got our reception here, right? Those two parts are all part of the reception. The cascade is when the first protein gets activated. Its job is to activate the next one. It's going to do that by adding a phosphate group. It took that phosphate group from an ATP, right? Then this one's going to go off and find another protein and give it a phosphate, not the same one that it has. That came from another ATP, because the ATP are just floating around all over the place, right? This can happen a number of times. There's no set limit. I'm sure there's some practical limit to this, but it doesn't really matter. For the examples that you might get, it could be any number of steps. The idea is that it's just like dominoes, where this one knocks down this one, which knocks down this one, which finally knocks down the thing that does the thing that we want it to do. It is all going back in reverse. Technically, there's proteins that are taking the phosphate off. That's what's happening in that particular image right here. Usually they're not going to ask you about that. But this is all just steps in a big like mousetrap type game. Now, sometimes the steps can be replaced or added onto by things called second messengers. These are important because they're not proteins. They're small molecules. A very common one is called cyclic AMP, or they could be ions like calcium. That shouldn't be a capital A, but whatever. Again, here's our diagram where we have to just follow the flow of information of the signal hitting the receptor, which triggers this thing to move, which triggers this thing to do something. In this case, this adenylyl cyclase is going to take an ATP and make it into cyclic AMP. Again, you shouldn't have to know exactly about that other than this is a molecule that could act as a second messenger. This will go off to set off the first protein kinase in that cascade. Or maybe there's only one protein in this case and it directly goes into the cellular response like this picture is showing. Or remember, there's no set pattern other than you have to go from one molecule to the next. So cyclic AMP is a small molecule and you can make a lot of them really fast. And that's something we'll talk about a little bit at the end of the video. Here's an even crazier version of a communication pathway, which is using calcium and actually another second messenger over here called IP3. There are a bunch of them. Again, you don't have to memorize them, but you do have to read the diagrams. So we follow the chain of the receptor triggering something, which in this case triggers something, which is going to, instead of triggering a protein, release a bunch of these little molecules. These molecules will then trigger something else to happen. In this case, to release calcium, and calcium itself is another second messenger. And as this part of the diagrams, I think a little bit more clear, you release a lot of calcium. Each individual ion can go off to activate something else, right? And so this is something we'll talk about later called amplification. You can get a lot of signal generated by using these second messengers, right? And so again, it's all just kind of this big chain. And so we made these out of paper during that lab. And so here was my version, just to show a general something that could have happened, where we have our signal molecule over here, and it hits the protein receptor, which changes its shape, which frees the purple one to go move because it used to be stuck to the protein, but now it's not. And that's going to turn on an enzyme, and the enzyme is going to change shape, of course, because that's the only way we can turn on and off molecules. And this one's going to take the ATP and make it into cyclic AMP, which activates the first kinase, which is going to take an ATP and give the P to the other kinase. And when you give it, it makes it change its shape, and that goes, and this just keeps going, right? And again, this could happen a bunch of times. It could only happen twice. Eventually, the chain leads to the response, and the response goes along and does the thing, right? So what is the response? The responses can be varied, of course, but there's two categories, cytoplasmic and nuclear. Cytoplasmic, the response is only going to reach something that already exists in the cytoplasm, probably a protein of some kind, probably an enzyme. In the nuclear version, it's going to end up all the way in the nucleus. This is going to be something we probably call a transcription factor, which will be the end of the chain, which causes the DNA to be activated and for transcription to happen to get the messenger RNA, which will make an enzyme, which will eventually go do something for the actual response, right? And so the cytoplasmic one over here, this one illustrates what I was talking about before, called signal amplification. If you follow along, it says that one molecule of epinephrine, which would be the signal molecule, is received by the protein receptor. But that's going to activate more molecules, more than one, 10 to the second molecules. And those are going to go off to eventually activate 10 to the fourth molecules, and then 10 to the fifth. And eventually you finally get 10 to the eighth molecules of glycogen that gets turned into glucose. This is part of the epinephrine pathway. But the point isn't what it's making. The point is that a lot of the steps, you can have one molecule that activates multiple molecules, and this gives you way more response to a very small amount of signal. The other part of the pathways is called specificity, and this goes back also to this picture. I might as well just go back to it. It's not that far. Oh crap, it is too far. I've already gone into this. I gotta just go. That's the one I was looking for, right? Where there's all these different versions of the pathways, right? It's kind of like a mix and match situation. So the signal specificity part, push the button super fast. There we go. Again, it's trying to show here that by interchanging the proteins that you make, right? Each cell has particular genes that are turned on for what it needs to do, which means it needs particular proteins that are going to do the things, which means it needs particular relay molecules most of the time. First thing to notice here is that all these signal molecules are the same, right? Same signal, but 5 different responses along the bottom. All of that is because changing either the receptor, right? If you change the receptor, you get a whole new pathway, right? Like totally different pathway. but some of these have the same receptor. But in one cell, the receptor, the available proteins are just these two, and that goes to the one response. But maybe in another cell that needs two different responses to happen, the receptor leads to this molecule, which has the ability to activate two things, and then you get two different responses. Sometimes you might want to have two necessary molecules. You want two signals. This one, you don't want it to accidentally happen somehow. So you need two different molecules to be created or received at the same time. That would then lead to activating this one very important protein that would go off and do the response, right? Again, you're not responsible for memorizing any of this. It's helpful to read the schematics, the diagrams that show you the signaling pathway. But every time you're going to have the reception, the transduction, and the response, right? And so there you have it. That's cell communication. And then more examples of when we will need cell communication will come up in the next video about the cell cycle. As always, let me know if you have any questions. And good studying.